thank you so much for the introduction, and thank you for having me here. It's my first time in a room with so many people for a long time, which is really exciting. I'm really glad to be here. So I'm going to talk about multi-omics and AI and multi-omics. And it occurred to me that I was put in the chemistry session, so I should say something about chemistry, but I am going to talk about metabolism, and I'm going to talk about aging, and I'm going to talk about AI. But I'll start with saying a little bit about multi-omics. So omics is something maybe in the biomedical sciences that has become a bit of a buzzword. But what it means is measuring a lot of things at the same time, so in different biological layers. So you have, for example, genomics, proteomics, metabolomics, and in each of these different layers, you're measuring a whole lot of proteins, a whole lot of transcripts, a whole lot of small molecule metabolites at the same time in the same sample. And from these measurements, we get an overview of the state of the system at a current point in time. And this is interesting because you have big data sets, really big, and these data sets are related to the same biological samples. So you can do all sorts of interesting questions, sort of asking, you know, what's the whole system doing together? But it also comes with many challenges for processing the data because the different measurements have different technical biases, different levels of resolution. You have different numbers of entities in these different. So you can quickly end up, if you're trying to do a study, you can quickly end up having some bias or noise or source of variation kind of driving your results and ending up with some false positives. And some of the challenges which come up in this kind of research have been and are increasingly being addressed by AI, without it being, I think, very necessarily well known. So for example, small molecule metabolites are often measured by mass spectrometry. This is a technology which lets you see in a biological sample which chemicals are in it, which specific structures they have, and the resolution for the whole sample of which chemicals are in it from the raw signal is often done with AI, and yet you don't see big billboards saying, well, AI driving your mass spectrometry. It's just part of the software that usually does these things. So I think there's a bit of a gradient between statistical sort of standard techniques that people are used to working with and then techniques which we call AI. And we call them AI when they're sort of new and doing something amazing that looks like magic. But then when they become run-of-the-mill, we forget that they are AI. And that's just something to bear in mind, I think. But yes. So the particular study that I'm going to talk about today was a study in C. elegans. I did my PhD in C. elegans in aging. C. elegans is a very famous model organism for aging research. It's a very small worm. And many of the things we know about aging at the biological level originally were discovered in C. elegans. It's quite easy to study aging in C. elegans because they go through their whole life cycle within 3 weeks. Quite hard to study aging in other organisms because you have to wait a long time for them to get old enough to be counting as aged. So in C. elegans, the main part of their sort of reproductive life cycle is over within about 4 or 5 days. And after that, they hang around, getting gradually more decrepit until they die at some point after 2 to 3 weeks of age in the normal case. And you can change this life dramatically. For example, if you give them really much too little food, then they live twice as long. In one of the most famous aging research findings ever, basically. Semi-starved creatures live much longer, which is really curious. And it has to do with how metabolism works. And sort of the opposite to if you eat a lot of sugar, you live less long. But anyway, what this study set out to do was to look at how the transcriptional changes, so really the expression of the genome in the organism, was changing at the same time as looking at how the metabolism was changing, in order to try and see whether we could discover new relationships between the gene expression, really the transcription of the genes, and the metabolism in the cell, because we knew that metabolism was what was driving differences in aging, in phenotypes. Health, not so healthy, etc. So the objective was to do this by having linked samples with which each sample then went for these two measurements, metabolome and the transcriptome. And then afterwards to put them together using various data analysis techniques which could put them together. But there are various challenges in putting together these different data types. So the study took place with 3 different strains. So in order to study aging in C. elegans, you need to use non-reproductive strains, because otherwise they have a lot of children, because C. elegans are self-fertile, they're hermaphrodites. And as soon as they start having these very, very small babies, it's quite difficult to keep the aging worms separate from the brand new young worms, and then you have difficulty in doing measurements. So we focused on wild-type-like non-reproductive strains, in order to have as close as possible to a regular, let's say, normal course of aging for this organism. And one of the first things we observed is that the transcriptome, so the gene expression, and the metabolome, so the actual levels of small metabolites in the worms, seemed to show very different drivers for their variability. So here you see the PCAs for the samples from exactly the same collections of worms. For the transcriptome on the right-hand side and the metabolome on the left-hand side. And what this visualization shows you is the variability in the underlying data projected into 2 dimensions. And you see that the strain is the main driver of variability. Even though these strains, the worms actually look the same and live the same length of time and in almost all respects. So it's quite a small difference, these 2 different ways of creating non-reproductive C. elegans. But from a metabolic point of view, the strain is not important. So the phenotypic difference that you see from looking at the samples is also shown much more closely in the metabolism. And in fact, what makes bigger difference for the metabolism is age. So what you see here is the transcription colored by age, where blue is younger and then red is older. And what the distance, so there's 3 strains in this image, and the space in between the dots is sort of close or further apart. So it's sort of a 3-dimensional extension of the 2-dimensional one we saw on the screen before. And what this shows you is that the transcriptome is mostly changing in the younger stages of the, mostly changing in the first part of, so it's mostly blue, this picture. The space is dominated by the blue and the red is all close together. And this is really interesting because in the metabolism, we saw exactly the opposite. So mostly the changes happen towards the end of the aging process. And some of the changes that we observed, so amino acids tended to decrease with age. You can see that because in this plot, the blue on the right-hand side, there's 2 strains, and decreases are shown as blue, while TCA cycle intermediates increase with age, so red is increasing. And one of the things which I think was quite interesting that came out of this study is that the time of day, because we were taking more than one measurement during the day in order to study short periods of time for the purpose of aging, we didn't intend to study time of day. But what we found was that for the metabolism, the time of day made a really big difference to the levels of certain of these metabolites, which were linked as well to aging. So what that means is that the time of day that you take your measurement for another study of aging metabolism, what you're studying may be impacted. I mean, this is probably well known to people who study metabolism routinely, but we observed it also with the worms. So then we faced the challenge of how do we integrate the transcriptome data, which was having these very different properties, and the metabolism data. And one way we came up with was using a kind of a pathway as an integrating model, mathematical pathway model, then taking the transcriptome data and using that to sort of change how much we represented in the model the enzymes availability, and taking the metabolomics data and using that with the time series that we had because we took several measurements in order to make predictions of how much of a given reaction was taking place at every point in time. And this is a method known as flux balance analysis, and it's one of the, let's say, more dynamic ways to integrate the transcriptome and the metabolome. And what we found when we did this was that if you take just the transcriptome, which is the kind of standard way to interpret, let's say, omics data with this type of mathematical model, then because the transcriptome is hardly changing later in the life of the C. elegans, you don't see much changing also in terms of the reactions. Because if you use a not so changing transcript to sort of control your reaction fluxes. But as soon as you add the metabolism, you see aging having dramatic changes on what's going on as soon as you add the metabolomics layer. So this was quite exciting because one of the things that came out of this joint multi-omics modeling approach was that the prediction was that one of the reactions that would be the most affected by age would be involving oxaloacetate in the TCA cycle. And of course, given that our prediction said that this should be a kind of a rate-limiting step in the TCA cycle as the organism got older, it was quite exciting to know that supplementation in C. elegans actually extends lifespan in older worms. So it seemed to be having some close relationship. But all of the analysis that we just did kind of depends on this representation of the pathways. And this is where I want to bring it back to AI. So in order to sort of analyze and interpret these data sets, we need a backbone of knowledge to take the data and sort of compare it to this backbone of knowledge. And so this is usually served in the biological domain, coming from all sorts of pathway maps, which are community curated, and ontologies and databases and all of this information. And what we discover sort of new, we feed back into these knowledge maps. But this process is kind of separated the data analysis side of things and the knowledge representation or modeling side of things. And it seems like there's a bit of loss or wastefulness there, where we could make this whole process work a bit more quickly if we could make more automation, get the knowledge back into those databases, and then use it in those analyses. So one of the things which I've been working on is trying to automate the step where you sort of get things in, because in our metabolomics panel, we had 110 metabolites that were measured, and of those, only 65 could match to the pathways. So there was massive loss there. And this is just because of different priorities. I don't think it's exceptional. I think it's quite typical that you cannot always match all your metabolites to pathways. And there is other, when you're trying to, I mean, the previous talk was also talking about data sort of matching and curation. So we're using machine learning to try to predict pathway and ontology annotations for chemicals. And I did talk about that last time in October in the network, and I'm not talking about it in any length now because of that, but I just wanted to say one thing, which was when we use the new transformer-based machine learning models, which is like, I guess, the cutting edge of AI at the moment, sort of at least of the deep learning part of it. Something really exciting, well, I think it's really exciting, is that when you look at the model, at the inside of the model, how it's working, you can see something like human-like understanding of the chemical language. So here you have what's called attention, which is a kind of a weighting of the input as the input is being processed for purposes of making predictions. And the attention comes in different layers, and the network is big and it has many layers. But these are just two of the selected layers. In this layer, it seems to be the attention is looking at the neighborhood. So it seems to be sort of learning the language. This is a smile string. It's a linear representation of chemical structures. And in this layer, the attention is looking at groups. So it's looking really at the chemical structural groups. And I thought that was really exciting, so I wanted to share it. So that's it. I'll stop there, and if there are any questions, thank you.